Good morning, dear students. In the series of lectures for microbiology, today we are going to discuss about the bacterial cell, structure of bacterial cells, specifically bacterial cell wall. For this lecture, we have in our studio Dr. Ramesh Kothari and Dr. Charmi Kothari, both from Christ College, Rajkot. Both of them, they have a good number of years for experience in microbiology as well as biotechnology, and both of them, they are heading the Department of Microbiology and Biotechnology respectively at Christ College. Now, we are going with the series of lecture regarding the structure of bacterial cell. It means we are going to study about the details of the bacterial cell, various components of the cell structure in depth. And to begin with, this is the first lecture in this series, that is the cell wall. Now, the cell wall is a very essential component of any cell, may it be plant or may it be protozoa or eubacteria. Well, in certain cases like animal cells, they do not possess the cell wall. Otherwise, most of the cells, they have an outer layer, a protective layer, which protects the cell from various functions, or which performs various functions and protects the cell from various chemicals or various other disturbances which are there in the environment. Now, this layer in case of bacteria is very specific because bacterial cell is a unicellular one and there is nothing else by which it can protect the cytoplasmic pouch which is inside the bacterial cell wall, and the cytoplasmic pouch contains the essential components for the cell's metabolism, and therefore it is one of the most predominant structure in the cell as well as an important structure. Now, this structure in depth will be discussed by two guest speakers, as I have told you, Dr. Ramesh and Dr. Charmi, in depth, and I hand over this mic now to Dr. Ramesh. Please, Dr. Ramesh.
Good morning, all of you. As we know that we are well aware with the statement of Darwin that survival of the fittest and struggle for existence. To be fit to survive, there are various protective mechanisms. And this protective mechanism is prevailing everywhere. If you see the world of tiny creatures, microbes, the microbes are surrounded by a protective layer which is broadly known as cell envelope, and this cell envelope, the outermost layer, which is known as cell wall. Before we go in detail about the cell wall, let me see some characteristic of a microbial cell. If you see this slide, the variation in the size of bacteria, if you see the dimension of bacterial cell, it is expressed in micrometer. Part of the cells are measured in nanometer. The average size of microbes is 0.5 to 2 micrometer. If you see the surface area, it is approximately 12 micrometers square. And surface area to volume ratio is 3:1. Food enters through the surrounding area of the cell very quickly because it is having a large surface area and reach to all part of the bacteria. Thus, this small tiny creatures having a largest surface makes this microorganism to survive under any conditions. If we think about if you see about the shape of bacteria, generally bacteria are monomorphic, but few are polymorphic. They change their shape as per the environment. They shows sometimes unusual shape, like star shaped bacteria, for example, belong to stela, and square shaped bacteria that are halo arcula. This is the slide shows the diversity among the microbial population. If you see this diversity in detail, generally the bacteria can be divided broadly into 6 categories as per their shape. Few are belong to coccus, some are coccobacillus, vibrio, bacillus, spirillum, and spirochete. Some are square shape, some are star shape. If you see the arrangement of the coccus bacteria, they arrange in various form. For example, when two round cell bacteria come together, coccus shape, it is known as diplococcus. When they arrange in a chain, which are known as streptococcus, and when they arrange in a clump, in a group, they are known as staphylococcus, and many times in group of 4 they are known as tetrads. As we know that there is a characteristic of each and every living communities. Likewise, there is a characteristic of this bacteria that they arrange in different fashions, and this arrangement of different fashions show their characteristics. If you see the bacterial structure in detail, the bacterial structure, if you divide this bacterial structure into 3 parts, that structure external to the cell wall, structure internal to the cell wall, and cell wall itself. So the bacterial cell wall is made up of a unique polysaccharide molecules which is known as peptidoglycan. It's a polymer made up of amino sugar and sugars. It is unique to bacteria. This peptidoglycan is not found in cell wall of any other bacteria, any other living system. It is having sugars like N-acetyl glucosamine and N-acetyl muramic acid. Generally, D form of amino acid use not L form. We know that L form amino acid are naturally occurring amino acids, but some unusual amino acid are found in the structure of bacterial cell wall, which are known as D form. It is very hard to break down the D form, and amino acid are cross-linked to N-acetyl glucosamine and N-acetyl muramic acid. If you see the characteristic of peptidoglycan, it is an unique macromolecules which is only found in bacteria, composed of a repeating framework of a long glycan chain, cross-linked by short peptides fragments, provides strong flexible support to keep bacteria from bursting because of change in the osmotic pressure. We know that bacteria are, if we see in nature, bacteria are encountering with various environmental stress. Many times the pressure, which is known as a turgor pressure exerted by the cytoplasm on the wall from inside, many times it makes bacteria to burst, but cell wall is the structure that protects this bacterial cell from bursting under some unfavorable environment. If you see the major difference between bacterial cell wall of gram positive and gram negative, wall thickness of gram positive bacteria are 20 to 80 nanometer, while in case of gram negative it is 10 nanometer. With respect to peptidoglycan we are talking about, the number of layer of peptidoglycan is 1, while in gram negative it is 2 to 3. Peptidoglycan covers more than 50%, while in gram negative it having 10 to 20%. Teichoic acid present in gram positive bacteria, which is not present in gram negative bacteria. If you see the peptidoglycan tetrapeptide in gram positive bacteria, most contains a lysine as a part of tetrapeptides, while in gram negative bacteria all contains diamino pimélic acid. If you see the peptidoglycan cross linkage in gram positive and gram negative bacteria, in gram positive bacteria generally pentapeptide, for example, entirely made up of glycine, while in gram negative bacteria the direct bonding between diamino pimélic acid, the third amino acid of one peptide, linked with the fourth amino acid of another peptide, that is D-alanine. The protein and lipoprotein It covers approximately 3%, and in gram negative it is around 50%. Lipopolysaccharide is not present in gram positive while it is present in gram negative. Gram positive bacteria are sensitive to penicillin while gram negative bacteria are not sensitive to penicillin. And if you see the lysozyme, your gram positive cells are sensitive to lysozyme compared to gram negative. Now if you see the picture of gram positive cell wall, you will find the mainly two layers arranged on the cell membrane, that is the periplasmic space and peptidoglycan. Now this peptidoglycan, it is an unique polymer, as I have already discussed, and gram positive bacterial cell wall contains teichoic acid. It is shown in, it is clear from the figure that the peptidoglycan matrix is cover the entire cell. Now the gram positive cell wall consists of a thick homogeneous sheath of a peptidoglycan which tightly bound the acidic polysaccharide, which include teichoic acid and lipoteichoic acid. This particular structure retain a crystal violet stain when we stain our bacterial cell wall during the gram staining procedure, because when we apply the primary stain, this primary stain, after application of primary stain we are applying iodine, and after that we are applying the decolorizer. So when we apply decolorizer, it dehydrates this cell wall, and the crystal violet iodine complex retained by the cell wall of gram positive bacteria, hence they color violet in color. If you see the strategy of gram positive bacterial cell wall formation, it's a polymeric chain of peptidoglycan stabilized by a cross connecting bridge of amino acids. Cross link form an interbridge, and pentaglycine peptides bridge are used. Generally, what happens? The pentapeptide or tetrapeptides, it is the characteristic of a species of, it is specific characteristic of bacteria. The covalently link D-alanine, that is the fourth amino acid of a tetrapeptide attached to the N-acetylmuramic acid with the L-lysine with third amino acid of another tetrapeptide chain. If you see in gram positive bacteria, generally in gram positive bacteria the third amino acid is generally lysine, while in gram negative bacteria it is diaminopimelic acid. And if you see in gram positive bacteria, you will find because the tetrapeptide chain is a stem polypeptide, and the interlink bridging polypeptide is generally made up of glycine. So this glycine, presence of glycine in gram positive bacteria, which is not the characteristic of gram negative bacteria, and this provide the strength to the cell wall, and this dense network protects cell from various forces. If you see the structure of peptidoglycan, as I have already discussed, it contains a unique alternative polymer made up of alternative polymer of N-acetylglucosamine and N-acetylmuramic acid, and you will find the peptide attached to the N-acetylmuramic acid, and it contains some unusual amino acid which are not present in the protein, and this unusual amino acid are synthesized by enzyme racemase from the usual amino acid. As we know that the naturally occurring sugar is D form and naturally occurring amino acid are L form. So from L form, this enzyme convert into D form for this special purpose. So if you see in this figure that tetrapeptide contains L-alanine, then D-glutamic acid, then diaminopimelic acid, and D-alanine. So this peptide chain, pentapeptide chain, sometimes it is a tetrapeptide chain, and this is the species specific characteristic, and many times used for the classification of bacteria. If you see this gram positive, you will see here that the cross linkage having 5 unit of glycine. Fine. This is the you will find in Staphylococcus aureus. This is again in detail. You will see the cross linkage, the bridge between two polypeptide the polypeptide molecules in peptidoglycan. This is again, you can see the alternative bridging between, you will see from the figure that there is an alternative unit of N-acetyl glucosamine and N-acetyl muramic acid. Now this N-acetyl muramic acid is attached with another N-acetyl muramic acid with the this tetrapeptide linkage. This is one ball and stick model of peptidoglycan. Now the unique characteristic of this gram-positive bacteria is teichoic acid. Teichoic acid is acidic anionic polysaccharide which contains carbohydrate like glucose, then phosphate and some alcohol. Now this particularly the acidic nature, because this teichoic acid bind with the proton, and binding with proton makes this particular environment acidic, and which prevent the cell from the action of autolysin. This teichoic acid many times bind with positive ion like calcium and magnesium, and this calcium and magnesium ion binding which useful for the identification of host by many bacteriophages. Under the limited supply of phosphate, or many times this teichoic acid replaced by teichuronic acid, and this teichuronic acid is the polymer of uronic acid and N-acetyl glucosamine. Now this teichuronic acid formation take under the limited supply of phosphate, because this limited supply of phosphate occurs because this phosphate is required by ATP and for DNA synthesis, and that makes again the environment suitable for acidic environment for the cell wall peptidoglycan. So two major forms of this teichoic acid I have already described, that ribitol teichoic acid which contain long polymer of ribitol, 5 carbon sugar alcohol, glycerol teichoic acid which have long polymer of glycerol and 3 carbon sugar alcohol. The sometime linked to the the plasma membrane lipids, that is lipoteichoic acid, extend through the peptidoglycan layer to the external surface, linked to the muramic acid of peptidoglycan via phosphodiester bond. And this teichoic acid, the function of teichoic acid is just like a weaving or a sticking the various layer of a peptidoglycan, which provide more tensile strength to the peptidoglycan, various peptidoglycan layers. If you see in detail if you see the lipoteichoic acid in diagram, it is connected to the cytoplasmic membrane, and teichoic acid, some teichoic acid are simply present in the polypeptide layers. Now the lipoteichoic acid which pass through this peptidoglycan layer and reach to the cytoplasmic membrane, this provide the stitching action, or it provide the you can say the intra layer peptidoglycan layers to form a, and it is just work as a thread like action. So this teichoic acid is the characteristic of gram positive bacteria. Now Dr. Charmi Kothari will discuss on gram negative bacteria, the cell wall of gram negative bacteria.
Yeah, Dr. Charmi, please be here. She will manage.
Good morning, friends. Dr. Ramesh Kothari has given you a brief description about the cell wall of gram positive organisms. I would like to give you about the gram negative. 3 words I would like to use for the gram negative cell wall. It's strong, it's tough, and it's elastic. Why we call it as a strong? The reason is that it can tolerate a high pressure. Why it is tough? Because it can, or it is strong enough to endure in extreme temperatures and pH. And elastic because it can expand themselves whenever it is needed in the environment. Molecular biology methods has not given any strong proof for the origin of gram negative cell wall, but the proof like fossils and other things showed that due to random mutation, selection and continuous change in the global environment has given rise to the 2 varieties of cell, that is gram positive and gram negative. First I would like to show in this slide a very clear picture about the cell wall of gram-negative organisms. This is an electron microscopic picture of a gram-negative cell wall. You can say it is a multi-layered picture. You can compare it with the gram-positive cells. You can see one membrane is extra over there. So simply whenever you read, you can see that gram-positive has a very simple structure and gram-negative has a complex structure. This complexity is due to outer membrane. In the next slide, you can see the three-dimension picture. You can see the base, the plasma membrane, above that a very thin layer of peptidoglycan, and the outer membrane. You can see in the outer membrane there are various components. First one, LPS, that is lipopolysaccharide on the left hand side, made up of two other things, that is O polysaccharide and lipid A. You can see on the right hand side that is porine protein, phospholipid is there, lipoprotein is there, and enzymes in the periplasmic space is there. This is a three-dimensional view. We'll see in a simple way in the next slide. Yeah, this is more clear. Just above the cytoplasmic membrane, you can see the peptidoglycan layer. Now you can compare it with the gram-positive cells. In that you have seen it was very thick. So gram-positive cells, it has a thickness of 20 to 70 nanometer, but in case of gram-negative cell wall, it's very thin, maybe 2 to 3 nanometer. Above the peptidoglycan layer, there is lipoprotein. It is just in the inner layer of the outer membrane. Now this lipoprotein, from slide you can make it out that it is just connecting the peptidoglycan layer with the outer membrane. Lipoprotein, lipo means fatty acids, protein. So fatty acid part of this lipoprotein is attached to the inner membrane of outer membrane, inner layer of outer membrane, and the protein, you can see it is bounded to the peptidoglycan layer. So in short, you can say that it is providing attachment. Fine. Above that, you can see in the outer membrane there is one extra layer called as LPS, lipopolysaccharides. Lipopolysaccharides are even called as endotoxin. The reason is that when this LPS is removed and when it is entered in an animus, it gives many shocks or fevers or death-like symptoms. Other things you can see is a protein channels, you can see, which is pink in color. That is acting as a porosity of this membrane, and other two proteins you can see it is there on the surface. So in short, this outer membrane is made up of 4 components. Next slide. Yeah, this is again the same picture, but in more detail in a 3-dimensional views. You can see with the different colors. You can see the O polysaccharides, or even it is called as O antigen, a core polysaccharide. It is all the component of outer membrane. So outer membrane composed of LPS, that is lipopolysaccharide, phospholipid, proteins, and enzymes. Sorry, next slide. Yeah, this is what gram-negative cell wall and outer membrane containing LPS, thin shell of peptidoglycan, periplasmic space, and the inner membrane. As sir has already told you about the gram staining procedure, that gram-positive cells get stained violet in color and the gram-negative red. The reason is that gram-negative cells cannot retain the grams iodine complex. So when you apply a decolorizer, it removes the primary stain, and when you apply the other stain, that is the counter stain, it takes the color of the other one. So you can differentiate on the basis of gram staining two different types of or two different colors of cell. Next. So that is what I was saying. Outer membrane composed of phospholipids, proteins, lipoproteins, and lipopolysaccharides. This is just an overall function of outer membrane, that overall the charge of the outer membrane is negative, which helps them for a defense in an immune response. Unlike the cytoplasmic membrane, the outer membrane is relatively permeable to most small molecules, that is due to the porine, which you have seen in the slide, which provides a channels to move. Next slide. That's what I said, porine, non-specific protein channels, barrier. It's a barrier to some low molecular weight compounds like antibiotic, lysozyme, and detergents. Now just we'll go in slide details of LPS, because it is one of the component which is responsible for the pathogenicity of many pathogenic organisms. LPS, it's made up of 3 parts: lipid A, core polysaccharide, and repeat polysaccharide. Repeat polysaccharide can even call as O polysaccharides or O antigen. Now lipid A, it is made up of 2 units of glucosamine, that is N-acetyl glutamic acid, N-acetyl glucosamine, and it is linked through ester linkage or amide linkage to the unusual fatty acids. It even contains a phosphate groups, and it even contains the phosphate groups which is being linked through this linkage. Now this part of LPS, it is responsible for the toxicity of as I told, it is called as an endotoxin. This part of LPS, it is embedded in the outer membrane. On the surface you have seen in the previous slide that it is being embedded. So out of 3 parts, lipid A is being embedded in the outer membrane. The second part of LPS, that is the middle part, core polysaccharides, it is again made up of sugars like NAG. Even it contains glucose, even it contains galactose, even it contains phosphate, but it contains even unusual sugars. Unusual sugars like 7 carbon containing sugars or 8 containing carbon sugars, heptose or KDO type of sugars it contains. That is the core part. And the next part is, in the next slide you can see it, that is the O antigen, or you can say it is a repeat polysaccharides, or it is even called as an O polysaccharides. Now in this there are 3 to 5 sugars. The common sugars which you can see is mannose, glucose and galactose. But along with these 3 sugars, it contains an unusual sugars, dideoxy sugars. Okay, so like paratose, colitose, abicose, these types of unusual sugars are there. Now these sugar units can be repeated in some case, and it can vary in the numbers also. So these are very specific to the gram-negative organisms. So that is one of the way for the identification also. So it can be used in a serology. It can be used for its identification because it's unique. It is unique among species and it is unique among subspecies also. Fine. As I told, LPS can be even called as an endotoxin. So what happens when human body come in contact with such organisms? Okay. Now what happens, what the body do when it comes in contact with such organisms? They first identify the microorganisms. Now how the microorganisms can be identified? All the microorganisms, they contain a specific unique molecules on them which is not common to the humans. So they identify these molecules. These molecules are called as PAMs, that is pathogen associated molecular patterns. These are unique to the microorganisms and our body try to identify it. Now these PAMs are sometimes even called as MAMs. The reason is that it's not only the pathogenic microorganisms which carry, even the non-pathogen microorganisms do carry, though they are not pathogenic. So we call it as MAMs, that is microbes associated molecular patterns. Fine. So what happens when, the previous slide, what happens when body come in contact with such unique molecules? They immediately bind to this. There are specific receptors called as PRR, pattern recognition sites, on the target cell of the defense system of the body. So when they identify it, they bind with each other and they trigger the innate immune system. Okay, like inflammations or macrophages and all that, and they start releasing the chemicals. So what happened during this phase, through the defense system, and through the defense chemicals, they will try to identify and they will try to remove it out. This is one character which is in favor of the humans. But sometimes what happens, even the microorganisms are smart enough, they try to play the sequence of the upper part of the they try to change the sequence of the O antigen. They change the sequence or they make it very unusual, so the host defense microorganisms cannot identify it. So in case of gram-negative cells, what are these PAMs? These PAMs are LPS, they are porins, and they are some of the fragments of peptidoglycan. So there are receptors, there are specific receptors to identify this part, they bind with this and they trigger the immune system, and they start doing or taking actions to remove these things from the human body. In next slide. Okay, this Ramesh sir has already explained you that as you have seen, it's a very thin layer of peptidoglycan joined with the third amino group to the fourth amino group of the nearby chain. That is what. Now one thing what he told that they have pentaglycine linkage. This pentaglycine linkage is never seen in case of gram-negative organisms. It's always a tetrapeptide. Fine. In the next slide you can see that. Okay. Next move. Fine. This is what the functions of LPS is, that it is providing a stabilizations to the outer membrane, and the O-side chain composition can be changed to avoid host antibody detection. That's what I told. Prove net negative charge on the cells. Prove net negative charge on the cells means it is carrying the whole negative charge. So whatever things, many of the things what comes in contact with them, they try to repel to each other. They will not allow to come in the cell. Next slide. Okay. Now lysozymes, cell wall can be removed easily with the help of lysozymes. There are many other ways also, but lysozyme is one of the biological way through which you can remove the cell wall. Now what this lysozyme do? They break the linkage between the two sugars, NAM and NAG. So once that linkage is broken, so you can say the compactness of peptidoglycan gets broke, and because of that the stability breaks, and when the cell wants to survive, or it cannot bear the osmotic pressure on that, so directly or indirectly the cells get dead. Next one. Okay, there are two types of cell, protoplast and spiroplast. Protoplasts are being found commonly from the gram-positive cells because it easily removes over there, and spiroplast, in that, you know, as you have seen, there is a complexity in the cell wall of the microorganism. So some parts remains attached to the outermost parts. So such type of cell we call it as a spiroplast. Next slide. Okay, before this slide, I'll just like to add few more things. You have seen in the second or third slide, there was a periplasmic space. Okay, this periplasmic space does a varieties of functions, like they will go for the oxidation reduction reactions, they will go for the osmotic regulations, they will go for the hydrolytic cleavage, they will go for the solid transport. So all that proteins, you have seen the porine proteins which are being allowing these things to go inside, and you have seen the other proteins. So other proteins which are there in the periplasmics do all these functions. One thing. Second thing, as sir told you about the gram-positive cell and the gram-negative cells, I would like to say gram-negative cells are less sensitive compared to the gram-positive cell. The reason is that the outer membrane which is there, it will generally allow all the hydrophilic molecules to come in, whereas it restricts the hydrophobic molecules. So because of that, what happens, whenever you expose the gram-negative cells towards the antibiotic, they will not allow the cells to cross the barrier. So that is the reason they are less sensitive and gram-positive are more sensitive. Now this is one other category of this. You can see the differences in the cell wall. You can see the differences over there. It has an extra mycolic acid. It's more commonly seen in Mycobacterium tuberculosis. So we exploit this property of the cell wall of acid first aid to recognize it. Next, in the next slide, you can see the comparison between the one which have the mycolic acid and the other which you don't have it. So you can see the comparison between them and how we can use it for the identification, or you can see the differences in the cell wall of both the organisms. This is a slide showing you the reactions, like gram staining reactions. What happens when the crystal violet stain is applied, like all the cells get or takes the color, the primary stain. Then when you apply a gram iodine, they form a crystal. Still all the cells remain colored. And when you apply a decolorizers, because as you have already seen the cell wall of gram positive and gram negative cells retain the primary stain, but gram negative, because of thin layer, they remove this gram iodine complex can get easily off from there, and when you apply the counter stain, it take up the color of the second stain. So you can differentiate two types of color, red and purple. Purple is for the gram positive and red is for the gram negative. So this is an overall function of outer membrane. It provides a protective barrier to the cells which is not there in the gram-positive cells. It inhibits or regulates the toxic compounds and metabolites, permits entry of small molecules through proteins, so indirectly making the cells safe from the external environment, and prevent loss of periplasmic enzyme. So because as I told, periplasmic enzymes does a varieties of functions, so this membrane helps them to make them remain in the cell. Next slide. So this is what overall function of the cell. It gives a particular shape to the cells and protect the cells from bursting. Along with this, there are other functions also, that it allows the transport of nutrients, it allows the it permits or it removes the waste material from the cell. But these are the two main important functions. What are the things which cause damage to the cell wall? One, lysozyme, as I told, they break the beta 1,4 linkages, and what happens when you treat it with the penicillin? Penicillin inhibits peptide bridges. So what does lysozyme do? It breaks the bond between NAM and NAG, whereas penicillin will break up the peptide bridge, that is between the two peptidoglycan layer. So in short, both of them are damaging the stability of the cell, or they are disturbing the compactness of the cell wall and indirectly disturbing the cell wall of organisms. Okay, this was all about the gram-negative organisms. Now what happens with the case of Archaebacteria? Fine. Archaebacteria, they do have the cells, but they do not glycan. Instead of that, they have the proteins and the lipids. But some Archaebacteria, they shows the property of pseudopeptidoglycan. In that category of Archaebacteria, instead of NAM, that is N-acetyl-muramic acid, it is being replaced by N-acetyl-talosamine uronic acid, and instead of L-amino acid, it has a D-amino acids. That is what a difference is in that. It is not in all cases of Archaebacteria. In fact, we can say that all the Archaebacteria doesn't show the same type of the structure. It is differing, as I have told, and sir has told about gram-positive and gram-negative. There is a variations in the Archaebacteria because some lives at a high salt concentration, temperature, pressure. So there is a difference. They make them stable according to the environment. And another one difference which I have read in the previous slide, here instead of beta 1,4 linkage, they have beta 1,3 linkage. Okay, so that is what the difference in the cell wall of the Archaebacteria. Next slides, please. And this type of properties you can see in some type of methanogenic microorganisms or halophilic microorganisms. As I told, they want to make them survive, they want to make them stable, so they change in their cell wall according to the need. Now this is what an overall picture of a gram-positive and a gram-negative cell. Now you can compare that. It is a simple one and another one is a complex one. Gram-positive is a simple one, gram-negative is a complex one.
So, as we discussed that we have compared the various structure and substructure of gram-positive bacteria, gram-negative bacteria, archaeal bacteria, which are known as extremophile, like methanogenic bacteria, halophilic bacteria, thermoacidophiles, and acid-fast bacteria. This all bacterial cell shows different pattern of their chemical nature. Though they have the same organelles, but if you see at the anatomy of that organelles, it shows some different chemical pattern. Now we know that for the classification point of view, nowadays we know that we are using the molecular techniques for the classification and categorizing bacterial cell. There was a method, and we are also practicing that method, that is known as chemotaxonomy. And this information, particularly the chemical pattern of different bacterial cell components, are useful, and it is very, very strong, and you can say the base on which we can rely. And as we already discussed, that certain group of chemical species are species characteristic of some selected bacteria, which is present in one bacterial cell, but it is not present in another bacterial cell. So this particular pattern of this chemical species we can take as a base for the identification and categorizing different bacterial communities. As we know that till date 1 to 2% bacteria are known and cultivated we know that till 98 to 99% bacteria we are not able to cultivate in laboratory, and for which we are relying on the structure DNA, which organisms which are not cultivable in bacteria. A very good emerging branch of science is coming, that is metagenomics, and that part is probably the future of various types of microorganisms.
Okay, thank you very much, Dr. Ramesh and Dr. Chami. They have presented a very in-depth lecture regarding the cell wall of gram-positive and gram-negative bacteria. Now, I would like to invite any questions from the students. If they have any doubt regarding this, we still have 10 minutes time where we can discuss these doubts of yours. So, I encourage the students, if they have any doubts, they can come up with it. Meanwhile, I would also like to add one or two points. One thing which is very unique about this bacterial cell, as you have seen, that peptidoglycan or lipopolysaccharide, whatever the composition is, they are basically a very large macromolecules. Then the question arises how these macromolecules are synthesized outside the cell, or how these structures they are made outside the cell. But let me tell you one point in very brief, that basically these structures, they are not synthesized outside, they are just transported from the cytoplasmic molecule to the cell, and where they are attached, where they are assembled. And for this, a very important role is played by certain carrier molecules like UDP, bacterophenol, which are responsible for taking these molecules. Synthesis of amino acids, synthesis of glucosamine structures, everything takes place inside the cell, that is the cytoplasmic pool. And with the help of these carrier molecules like bacterophenol, they are being transferred to the outer side of the cells, where the assembly takes place. So this is a highly intricate and complicated mechanisms which bacteria shows that even the large complex macromolecules can be made or can be used for the protection of the cells. Thus, to sum up, we can say that the cell wall is something like a very strong fortress, which in old days kings used to build surrounding the city, where the public or the citizens would be protected from any invasions. Similar to that, these cell walls, they protect their vital components which are present in the cytoplasm. They perform not only this protection functions, but apart from that, as you have been listening, they provide the shape to the cells. They are also the site of phage attachment. Then they are also responsible for imparting a specific antigenicity to the cells. We call them as an O antigens. That is the somatic antigens, which is due to the presence of these outer membranes, the cell walls. Moreover, they also help in serological identification of the cells, because when we have to identify the organism, basically a microbiologist, if it doesn't get any clues, then he is a lost person, because there are innumerable number of microorganisms, and he has devised number of ways to identify it. As Dr. Ramesh said, that the chemical composition of the cell wall also plays an important role in identification. So not only serological identification, but there are also the identification made on the basis of phage attachment sites. Bacteriophages, they also have a particular preference for a particular site, particular chemical groups on which they will attach, and then they will carry out their penetration of nucleic acid inside the cell. Likewise, it also has other function which is performed, that is to prevent the loss of enzymes which are present in a periplasmic. Periplasmic space is full of enzymes which are responsible for ATP generation, which are important for various vital functions of the cells. Thus, in a way, you can say cell wall performs so many important functions that a cell cannot afford to lose it. However, in certain cases, as it has been earlier mentioned, that with the help of certain enzymes or in presence of certain antibiotics, we are able to make the cell wall, or we are able to lose the cell wall, or we are able to see that the cell wall is detached from the cell. But then under those conditions, the cell only survives if the solution and other things or the environment is isotonic, which does not permit the cell to shrink, nor it permits cells to burst. So these are in short various functions which have been explained to you in very detail by Dr. Ramesh and Dr. Chami. Dr. Ramesh, you have anything to add to these things?
I would like to add something.
Yeah,
last.
please go on.
We know that Gujarat is known for pharmaceutical industries. While we all are behind pharmaceutical industries, we know that pharmaceutical industries are producing a lot of molecules which target the cells. And as we know that there are four main targets of any medicine. Yeah. That is the inhibition of protein biosynthesis, inhibition of DNA replication, inhibition of cytoplasmic membrane, and you can say inhibition of cell wall biosynthesis. So the pharmaceutical industries are first of all targeting the first, you can say if a particular fort is there which is surrounded by four protective layer. So very first layer is cell wall. Right. If you are able to break this cell wall, then everything is possible. Fine. So the cell wall is the structure. Knowing the detailed chemical structure of cell wall, pharmaceutical industry can devise novel target molecules which can reach to the pathogenic microorganism. We know that when pathogenic microorganism is invading, so if we start from the mouth to complete the entire intestinal tract, it contains more than 1,000 type of microorganisms.
Correct.
And along with this, more than 1,000 microorganisms or pathogenic microorganisms are also there. So knowing the detailed structure of cell wall, it will be useful for industries associated with pharmaceuticals to control the pathogenic microorganism particularly.
Yeah, Dr. Kothari very rightly said that if you break the first line of defense, then it is easier for organisms to, or for to control these organisms which are basically pathogenic. Therefore, the knowledge of cell wall is very, very important, not only for gram-positive or gram-negative organisms, but there are certain emerging diseases where the organisms, we have been able to look into those organisms for the first time. They may have certain different kinds of composition. And don't forget that the organisms, they always have an ability to mutate, that is to bypass the various aggressive role of an antibiotic or a chemicals. So it is a constant fight between microorganisms as well as the scientists to control them by using various chemicals or drugs or medicines or antibiotics. Thank you very much, Dr. Ramesh and Dr. Chami. I am highly obliged to you people because you have accepted the invitation and come to the studio to deliver a lecture. I know it is very difficult to prepare and spend such a lengthy lecture in such a perfect manner. So I personally thank you, as well as I would also like to thank the Director Higher Education who has initiated this program so that the live lectures reach to the different students across the Gujarat for various subjects. I am also thankful to Dr. Kamal who has taken initiative to carry out and coordinate the BISEC lectures, SANDHAN lectures. And last but not the least, I am also very much thankful to the various crews, the staff of BISEC studio who have remained behind the curtain but have been able to bring the excellent quality of this live coverage for the students. Thank you very much. Namaste.